Hello everyone, it's Kenji and we're back again with the pharmacology course. In the last video, we've made an intro to what is pharmacology and some basic concepts that are related to pharmacology like pharmacokinetics and pharmacodynamics. We started the last video with pharmacokinetics and the four basic concepts that are related to pharmacokinetics or the concepts that do actually represent pharmacokinetics. They are abbreviated in ADME as we've said in the last video. A stands for absorption, D stands for distribution, M stands for metabolism, and E stands for excretion. We started with absorption in the last video. Specifically, we talked about the factors that do affect absorption, and today we're going to complete the absorption part. And we did have a part that was missing in the last video, which we're going to explain in this video, which is the mechanisms of absorption. So without further ado, let's get started. Absorption of drugs. Absorption of drugs actually happens with four different mechanisms. The first one we're going to talk about is passive diffusion. What is passive diffusion? Passive diffusion means that the drug or any substance moves from a higher concentration to a lower concentration. The very normal diffusion. Any substance that diffuses without the need of any energy, it diffuses from a higher concentration gradient to a lower concentration gradient. We call that it's moving with the concentration gradient. Moving from a higher concentration to a lower concentration. So let's write that. Passive diffusion. No energy needed for passive diffusion because you don't really need energy when you're moving from higher concentration to lower concentration. No energy needed. And the second thing is moves from higher concentration, from high concentration to low concentration. So this is actually what's happening. And also passive diffusion mainly happens for lipophilic drugs. Could it happen for hydrophilic drugs? Yes, it could, but let's first explain why or how it does happen for lipophilic drugs. Let's just write that. Mainly for lipophilic drugs. All right, let's use a different color. So this one here in the orange square is a drug. This D stands for drug. And the orange ones are lipophilic. Okay, let's do the orange. Lipophilic. They dissolve in lipids, which mainly means that they're going to dissolve in the cell membrane layers because as we've mentioned in the last video, the cell membrane consists of exactly a phosphobilipid layer, which is a lipid. So like dissolves like, so lipophilic substances are going to dissolve in lipophilic materials like the phosphobilipid layer in the cell membrane. So we have a lipophilic drugs. So what's this area that they're found in? They are found in the extracellular area of the cell membrane. Cellular area of the cell membrane. This is where the drugs are initially found before getting absorbed inside the membranes of the body. Let's do this to make it clear. So, and we have here our lipophilic drug, our lipophilic drugs, and this is the cell membrane. Okay, and this is the cytosol, which is already inside the cell. We've mentioned before, and we've agreed that lipophilic substances or passive diffusion moves from higher concentration to lower concentration. So this means that the concentration of this drug outside in the extracellular area is high, and it will move inside the cell through the cell membrane. So it will move easily, very easily, because it's lipophilic as the cell membrane, which is a phosphobilipid layer. So it doesn't need any kind of transporters, any kind of carriers, as we're going to mention in the other mechanisms. No, it's going to pass very easily through the cell membrane because it's a lipid, and the cell membrane is a phosphobilipid layer, so like dissolves like, so it's going to pass very easily. And through the diffusion, we're going to have the drug particle inside the cell through passive diffusion. This is how lipophilic drugs mainly diffuse through the cell membrane. We don't need any kind of carriers, as I've mentioned, just from a higher concentration to lower concentration, and they enter the cell. Very easy. We mentioned that passive diffusion mainly happens for lipophilic drugs. Could it happen for hydrophilic drugs? Yes, but under very limited factors, actually. It could happen for hydrophilic drugs, if they are the following. Number one, very small. Number two, non-charged. And yeah, that's basically it. So if you have a hydrophilic substance that's small and non-charged, why non-charged? If you watch the last video, we mentioned that the cell membrane, which is a phosphobilipid layer and has a phosphate group is charged. So it won't accept any other charged particle because this will cause repulsion, and repulsion will not help with the absorption process. So you need the substance that's going to be absorbed through the phosphobilipid layer and the cell membrane to be non-charged. So any hydrophilic molecule that's small and that's non-charged and has a higher concentration of the drug outside the cell than inside the cell could be absorbed through passive diffusion. But would it pass through the phosphobilipid layer directly like lipophilic drugs do? No. It will pass through the cell membrane, but through certain kind of channels, like this one. This is a hydrophilic drug. Dividing the words into syllables because of the space. That's okay. This is a hydrophilic drug. It's going to pass if it's small, as we've mentioned, and if it's non-charged, with a reason we've mentioned as well, it's going to pass through a certain channel to get to the cytosol, to be finally inside the cell. So it's through passive diffusion because it has all the following factors or all the following conditions that's needed for passive diffusion to happen, but not exactly in the same way that happens to lipophilic drugs. Lipophilic drugs do not need any channels or anything to pass through the cell membrane. It passes directly through the phosphobilipid layer that makes the cell membrane. But hydrophilic drug cannot do that because they're not lipids. They're not lipid soluble. So they're going to pass through a certain type of channels to be finally, as we've mentioned, inside the cell. So that's for passive diffusion. The second type is facilitated diffusion. Facilitated diffusion, there are some common things between facilitated diffusion and passive diffusion. The first of them, facilitated diffusion, doesn't need any energy. No energy needed as well in facilitated diffusion. All right, that's good. And there's another common thing, which is it moves from high concentration to low concentration. So until now, we have two same conditions between facilitated diffusion and passive diffusion. So what's different? It happens for large charged, either large or charged. It doesn't have to be both, all right? Hydrophilic drugs. We mentioned here that for a hydrophilic drug to pass through the cell membrane with passive diffusion, you need to have all of these conditions. No energy needed from high concentration to low concentration. Both of these things are common in facilitated diffusion. And it needs to be small or non-charged, or both. It needs to be both, actually, both small and non-charged. But if this hydrophilic substance or drug is either large or charged or both, it won't pass through the cell membrane with passive diffusion. And passive diffusion also means actually that it does pass through the cell membrane without the need of any type of carrier or transporter. For lipophilic drugs, it passes directly through the cell membrane, and for hydrophilic drugs, it passes through a channel. But a channel doesn't mean a carrier. But in this case, in facilitated diffusion, it does need a certain type of transporter, which is this pink one. That's a transporter. So what actually happens here? Oh, I forgot something. Yeah, I'm sorry. I need to draw this. I need to complete it. Can't be looking like that. Yes, almost there. Perfect. Done. All right, so that's our drug that we agreed it's either large or charged or both. And it's hydrophilic. Lipophilic drugs only pass through the cell membranes with passive diffusion. Just put it in your mind. If the drug is lipophilic, it's going to pass through the cell membrane with passive diffusion. That's it. All the other mechanisms are mainly for hydrophilic drugs. So here's our drug. It's going to be here. This is a transporter, as we mentioned. It's very large, so the cell membrane will refuse it. I won't get you inside. I won't get you. You're hydrophilic. Yeah, you won't fit inside this channel to pass through. You're charged. Yeah, the phosphate groups will repel you. So we need to find another way for you to enter the cell membrane. And this other way is the transporter. So the drug gets inside the transporter and the transporter makes the job. How does the transporter make the job? This transporter makes conformational changes to its shape. With these conformational changes actually do not require any kind of energy. When it makes these conformational changes, it allows the drug to be inside the cytosol. Inside the cytosol means it passes into the cell and be inside the cell after it passes the cell membrane. So here's our drug. And it's finally, with the help of the transporter, inside the cytosol. So that's facilitated diffusion. It happens for hydrophilic drugs that are either large or not, or charged, or both. Now we move to the active transport, and I think you've heard it before. It's pretty familiar. Which color should I? Yeah, the black one. This is the first mechanism of absorption that we're gonna need energy in. We're gonna need ATP in. So let's write in. Energy is needed. We need ATP in active transport. Just to highlight it. Energy is needed in active transport. And why energy is needed? What makes him so different from facilitated diffusion and passive diffusion that didn't require any energy? It moves from lower concentration to higher concentration. So it doesn't move with the concentration gradient as passive diffusion and facilitated diffusion do. It moves against the concentration gradient. So it moves from lower concentration to high concentration. That's against the concentration gradient. That's not normal. So when something is not normal to happen, it requires energy, because the normal path is for things to pass from higher concentration to lower concentration. When you want to make things go from lower concentration outside the cell to a higher concentration inside the cell, you actually need energy. So that's why active transport exists. So let's explain it. Here we have our drug. All right, that's the drug, and that's a carrier. All right. Okay, let's make better demonstration. Let's say that this is a carrier. All right, and we will have our drug going in here to be on this carrier, and we've agreed that this drug is of low concentration. Here we are in the extracellular area. So we have inactive transport, we have lower concentration outside, okay, no problem, and higher concentration inside. Let's write it. This is the cytosol inside the cell, and this is of high concentration. Wow, that's okay. This is a high concentration. So this carrier will use ATP. Let's write ATP with a different color. We'll use ATP, and we'll get us ADP. That's not really significant, okay. And we'll make the drug get inside. And that's how it's done. Bingo, we're here. So active transport happens when you're trying to move a drug from a lower concentration to a higher concentration. So this is against the concentration gradient, because this requires energy. And this also happens mainly for hydrophilic drugs. We've agreed here that a lipophilic drug doesn't need any mechanism other than passive diffusion. Actually, you might ask yourself, what if there's a lipophilic substance or drug that has a lower concentration outside the cell in the extracellular area and a higher concentration inside the cell? Wouldn't that require active transport, for example? Active transport, as we've mentioned here, and as in the diagram, requires a carrier, or facilitated diffusion requires a transporter. Lipophilic substances or drugs do not usually have transporters or carriers for them on the cell membranes. Carriers and transporters are mainly for hydrophilic drugs. So actually, theoretically, if we do have a lower concentration of a drug, a lipophilic drug, outside the cell membrane and a higher concentration of this exact same substance or drug inside the cell membrane, passive diffusion will still happen. How? The drug will move from inside to outside with the same exact mechanism. So it doesn't have to get inside, it will get outside, but it will keep the same mechanism, passive diffusion, because you don't need anything else. You don't need it. Lipophilic is lipid, so it dissolves in lipid, substance like dissolves like. So you don't need any energy, or you don't need any other mechanism. So in this case, if it's a lower concentration outside and a higher concentration inside, it will be reversed. The drug will move from inside the cell to outside the cell. So that's it. Lipophilic drugs, passive diffusion. Remember that. So yeah, let's write anything else we need to write here. Yeah, mainly for hydrophilic drugs as well. Mainly for hydrophilic. Right, and the last one is endocytosis or exocytosis. Endocytosis, actually, let's write it first, requires way more energy. I'll write it in the same way more energy than active transport. Why? Let's see here. Let's first explain on the diagram and you will know why endocytosis or exocytosis require more energy than active transport. Here we have drug particles. Let's first write something that exocytosis actually does happen for excessively large molecules or drugs. The molecules that are excessively large get absorbed through endocytosis or exocytosis. Endocytosis mainly for absorption, and exocytosis is just the reverse of the mechanism, just to be more specific. So here we have the drug particles. This is our drug, and as we agreed, This is the extracellular area. This is the cytosol. And this is the cell membrane. All right, this is the cell membrane. This is our drug. When the drug is excessively large, it doesn't go through active transport or facilitated diffusion. No, it goes through endocytosis. The cell membrane starts to fold itself, as you see, like here, starts to fold to form a vesicle at the end. This is called a vesicle. This vesicle holds the drug molecules inside of it to be then used in different things inside the cell. As for example, the things that get absorbed via endocytosis, let's say, is vitamin B12. Vitamin B12 gets absorbed in the same exact mechanism. And why endocytosis requires way more energy? Because if you thought about it logically, active transport has a carrier. All what the carrier does is that it gets the drug on it, the hydrophilic drugs, and then when the drug gets on it, it uses ATP, and then by the help of ATP, it helps the drug to move from lower concentration to higher concentration against the concentration gradient to be inside the cell. But what happens here is that the cell membrane itself folds around the drug particles, folds around it in steps until it forms a vesicle around many drug particles. This mainly requires way more energy than having a carrier that will use ATP, one ATP for each molecule, to get the drug inside. Making the cell membrane fold around itself and fold around the particles of the drug to form a vesicle to get inside, to get this drug inside the cell membrane, logically requires way more energy than getting one molecule inside using a carrier, because the cell membrane does the job itself here. It doesn't require a carrier. By endocytosis, there are no carriers. The cell membrane itself is the one that folds around itself and folds around the drug particles to move the vesicle that will give the drug inside the cell membrane. So that's why it requires way more energy than active transport. And it does mainly happen for proteins. Let's write that. Peptides, some substances like, let's first write the example, vitamin B12. Okay, this is by endocytosis. Endocytosis. And other examples for exocytosis, for example. Exocytosis is just the reverse of this. You have a vesicle and it goes like that, until it gets out of the cell membrane vesicle. And examples for exocytosis also will be norepinephrine. You know norepinephrine inside the synapse of the neurons? When you get calcium influx inside this synapse, and then the vesicle that has norepinephrine inside of it gets released, and releasing all the norepinephrine, this is exocytosis. So norepinephrine, and we're going to get into that in details as well, going through the pharmacology course. Norepinephrine by exocytosis. These are all examples. And yeah, we mentioned here that it could happen for proteins, peptides, peptides, and other substances that are excessively large, as we've mentioned here. So this is mainly all the mechanisms that absorption inside our body happens with. And yeah, I hope you got some beneficial information in this video. This is mainly all about the absorption part in the pharmacokinetics. In the next video, we'll get to explain distribution, then metabolism, then excretion, to finish all the pharmacokinetics, and then get continued with our course. So this was our video for today. I hope you enjoyed the video, and if you have any feedback, please leave it in the comments. And thank you for watching the video. This was Kenji, and have a great day.